Welcome back to Pre-Health Shadowing, everyone. My name is Nina, and I am the founder and CEO of Pre-Health Shadowing. Here for another shadowing session, we are really honored to welcome you all. Pre-Health Shadowing is a student-led, women-led, nonprofit organization focused on creating flexible and accessible learning opportunities for pre-health students worldwide. We do have closed captioning to accommodate students of all abilities and needs. If you have any ideas about how we can be more accessible, please email us at info@prehealthshadowing.com. Additionally, if you'd like to stay in the loop, you can save prehealthshadowing.com as a contact on your email to avoid going to spam or the promotions folder and send it straight to the inbox so that you can register for your virtual shadowing sessions. You can join our email list on our website, prehealthshadowing.com. Additionally, we are on social media, so if you are too, you can give us a follow on Instagram, TikTok, and Facebook at Pre-Health Shadowing. Since we are a global organization, I want to know where you are Zooming from today. Feel free to drop it in the chat. I'm currently calling from California in the United States. If you are interested in being a part of pre-health shadowing, we do have opportunities for students just like you to be a part of the pre-health shadowing team. We have various opportunities depending on your availability and what you're looking for. We have opportunities in volunteering where you can make your own schedule, volunteer on your own time for a minimum of 1 hour per month, and be a part of our mission. We also have leadership opportunities if you're looking to be a more integral part of pre-health shadowing. You can apply on our website, prehealthshadowing.com. If you go to the volunteering tab, you can sign up to be a volunteer or apply to be a leadership team member. If you have any questions about this, do stick around for the end of the session. We'll be opening up for questions. If anybody has any questions they'd like answered, I'll be here and I'll be able to chat with you about our opportunities. Some of the roles include and are not limited to IT, if you're into tech, grants and fundraising, if you're into finance, video productions, if you're into media studies, research program, if you're into coordination or research, and more, featuring diversity and inclusion, recruitment, volunteer communications and coordinations. All of this, you can find more information on our website below, prehealthshadowing.com. If you're a high school student interested in getting involved, we do have a high school program as well titled HTP. HTP is a group that works to establish pre-health shadowing as a club on various US campuses. So if you're interested in establishing pre-health shadowing as a club on your high school campus, join the team and get started as a pre-health shadowing club ambassador. If you're interested in getting published, we do have a blog where we're hosting individuals' pieces of work in the pre-health shadowing community. You can submit your articles, reflections, reviews, and success stories to prehealthshadowing.com/blog submissions. Like I mentioned, we are a nonprofit organization and run solely off of the time and donations of our volunteers and our community. We do ask that if you are able to donate, please scan this QR code and help us out. We want to run a free organization. However, running an organization is not free. And so we will never charge individuals for the services that we provide. All that we ask is that you can please send this to members of your community, members of your family, to help keep pre-health shadowing accessible to everyone. Throughout the duration of today's session, I invite you to type your questions in the chat. We are honored to be given a presentation from a professional today, and now is your chance to get your questions answered. So please utilize this chat for any questions that you may have. We will ask them during the second half of today's session during the Q&A portion. I encourage everyone to take good notes because at the end of the virtual shadowing session, there will be a post-shadowing assessment. This post-shadowing assessment is optional, but if you're interested in earning a digital certificate to verify your hours with pre-health shadowing, you can take the post-shadowing assessment. If you get a 70% or higher, you are eligible to receive a certificate. If you are able to, I invite you to turn your cameras on. While this is not a requirement, it just makes a great environment. Unfortunately, today we did have some technical difficulties with our professional, and so our professional will be unable to share their camera. However, if you are able to share your camera, I invite you to do so. Without further ado, I'd like to welcome to you our professional for today, Dr. Raymond Roos. Thank you so much for joining us. I'm gonna let you go ahead and share your screen whenever you're ready. And for the students that are in the Zoom call, I'm gonna go ahead and launch a poll. Please take a few moments to answer these questions. They're very general. Where are you in your journey? High school, undergrad, graduated? Where are you calling from? What are you interested in? And a few questions about PHS. So go ahead and take a few moments to answer that, and then we'll jump into our virtual shadowing session today. Awesome. If you haven't yet submitted the poll, go ahead and do so. Alrighty, I'll be closing it in just a few moments. And I'll share it with everyone so everyone can take a look around the room. All right, I'm going to end it and share. So as we can see, we have a lot of students in their gap year, so looking to get some insight into different health fields. We have students calling from the West Coast, West Coast represent, that's where we're at. Lots of students interested in getting a medical degree, MDGO. We got a couple potential RNs, PAs in the house. Some interested in research. Welcome to those of you who this is your first virtual shadowing session. And if you're interested in volunteering, I will go ahead and send the website in the chat so that you can check out our opportunities on there. During this time, I'd like to welcome to you our professional, Dr. Roos. You may go ahead and get started whenever you're ready.
Oh, thanks, Nina. Can you hear me okay?
Yeah, we can hear you.
First, it's a pleasure for me to provide this presentation. I hope you like it. The title is a little complicated, but you'll get the gist of it. The path from New Guinea cannibalism, the mad cow disease, to all neurodegenerative diseases. On the left side, there's a picture of the University of Chicago, and I am a professor in the Department of Neurology at the University of Chicago Medical Center. On the right side is a statue of a cow, and they had a kind of display of cows throughout the city, and this is just one example. So somehow, the second slide, and I want to make one point here that I want to make on this slide and the next one, and that is the role of the mentor. Everybody needs a mentor, somebody who's going to guide you, give you inspiration. Maybe you're going to have more than one mentor. I had two mentors. The first one being Stanley Aronson, who was the head of neuropathology at Downstate Medical Center, and then became the dean at, I think it was University of Rhode Island Medical Center. and he took a certain number of medical students and provided some of them with summer fellowships. And I had a summer fellowship at the National Hospital in Queen Square, London. And this on the right side lower is kind of an old picture of the wards there. But I could tell you when I was there was a big room with 20 beds, and each bed had a curtain that could go around it. So it was a very public clinical space, no question. And the National Hospital was the premium neurology in the world when I rotated there during the summer. And there were just astounding people there. And it really fulfilled my interest and desire for neurology. And here's my second mentor, which was a man called D. Carlton Gattashek, who probably nobody listening has ever heard of, but Gattashek won the Nobel Prize. And between my medical school and my medical internship, I spent 2 years at the National Institute of Neurological Diseases and Stroke at the NIH, working for Carlton Gattashek. And you see him kind of surrounded by all of these boys from Melanesia. And I'll get into the story. But it's an important story. Important story because Gattashek won the Nobel Prize, and another individual, Stan Prusner, won the Nobel Prize for these diseases. Not that they're all that common, but that they had a very important impact on our understanding of biology, medicine, and science. And Gattashek did his investigations in Papua New Guinea. And you see here a map of the world and this little island off the coast of Australia, which is this big landmass here. And Papua New Guinea, when Gattashek first went there, was a wild area. He had been stationed in Australia, and he had his base in Okapa in Papua New Guinea. And there he saw children in the island that were dying of a disease that probably no one on this PowerPoint presentation has ever heard of, a disease called Kuru, K-U-R-U. And there was really very few people that were interested in this disease in New Guinea. One of them on the right is Vinzigas, and Gattashek is the person who's got his hand on the abdomen of this child. And this was the 1950s. And Gattashek was working for the Walter Reed Army, a man named Joe Smardell. And here he's writing a letter, March 1957, from Okapa. I am in one of the most remote, recently opened regions of New Guinea in the Eastern Highlands. And there's no question about this, by the way. In this each other, as of a few days ago, and cooking and feeding the children the body of a Kuru case, the disease I am studying. nobody knew the cause of this disease. And as Gadushek says, it's so astonishing an illness that clinical description can only be read with skepticism. And I was highly skeptical until 2 days ago when I arrived and began to see the cases on every side. So as I mentioned, he's stationed in Australia, working in the lab of actually another Nobel Prize winner when he heard about these cases. And Gadushek was a world explorer. So he continues the letter. Should you be able to make a grant? So Gadushek needed money. Everybody needs money who's in science. Based on little more than your assurance of my integrity and medical judgment, I tell you, Joe, this is no wild goose chase, but a really big thing. Everything in my medical training makes me confident. Let me know so that I can plan to extend my work here for at least several further months. On my own, I can hold out for 1 or 2 months and still have enough to get home via Europe. But I am fully aware of the unwiseness in trying myself to be both investigator and philanthropist. Enough said. I stake my entire medical reputation on this matter. This class of illness is not my specialty, but I can handle research on it as well as anyone. So it's important to know that Gadushek was trained, he was an MD, trained in infectious disease, actually with Enders, who won the Nobel Prize for poliovirus work. But nobody quite knew what was going on with Kuru. And this shows a part of the brain called the cerebellum in a normal individual. And on the right side, Nina, can they see my pointer?
No, I don't think we can see
Oh, okay. On the right upper side is a Kuru cerebellum. And there's 2 things to note. And one is that the density on the left side is no longer there. In other words, a lot of cells are lost. And then the other point is there are these little large cells between the dense cell population and the less dense cell population, Purkinje cells, and they are lost in the Kuru cerebellum. And interestingly, under high magnification, the neurons are vacuolated. So the next clue to these diseases comes from a man named Bill Hadley, who was a veterinarian neuropathologist. And I have this cut off a little bit. I'm trying to, whoops. At any rate, I can't read that top line, but he says it's difficult to make analogies between diseases of man and lower animals. And it's attended by numerous pitfalls, many valuable clues, contributing to the understanding of the fundamental nature of the disease can be gained from a broad comparative viewpoint. With this in mind, I should like to present a few brief comments on the similarity of 2 progressive neurodegenerative diseases of the central nervous system, namely scrapie, which is the disease of sheep, and Kuru, affecting, it was one particular area of New Guinea in which the Foray natives lived, and the Foray, as Gadushek mentioned, were cannibalistic. And they practiced something called endocannibalism. They ate their own as a way of mourning their own, and they gave the brains to children and women, adult women. And Hadlow says, I do not suggest that these diseases are identical or even counterparts, but in my opinion, their overall resemblance is too impressive to be ignored. Despite the lack of indication suggesting that scrapie is an infectious disease, the disorder can be induced experimentally in the sheep and the closely related goat. Thus, it might be profitable in view of veterinarian experience with scrapie to examine the possibility of the experimental induction of kuru in a laboratory primate. But one surmised that the pathogenetic mechanisms involving scrapie, however unusual they may be, are unlikely to be unique in the province of animal pathology. So what's important to know is that scrapie was a chronic disease, and kuru also. Individuals with kuru lasted months, 6 months, a year, something of this sort. So these weren't acute infections like we normally know. And Gattashek read Hadlow's comment, and this shows this kuru up on the left upper neuron that's vacuolated, and then scrapie with neurons of scrapie that have similar vacuolation. And you could understand how Hadlow saw the similarity of these diseases. And then you see a sheep that's scraping its fur on this pole, which is the reason it's called scrapie. These animals have abnormalities in sensation and scrape as a result of it. And this was a phenomenal experiment that Gattashek did, in which he collected central nervous tissues from a number of kuru victims. And basically, I'm not saying I'm promoting this in any way, but he traded brain tissue for various things that the islanders wanted. And as a result of that, he was able to collect a bunch of brain tissues and ship them off. By that time, he was in the National Institutes of Health. And each one of these, on the left side, the upper rectangles are kuru victims. And the line going down is the experimental inoculation. And although it might be difficult to see, these animals were fine unless one started to look months later. The average incubation period of kuru in a non-human primate was something like 18 months. And the brain tissue of these non-human primates had enormous vacuolation. So Gattashek was excited about the transmission of kuru. It meant that this disease that had no inflammation and was really chronic was a transmissible disease. And so he sent tissue to Igor Klatso, again, 1950s. And he said, Klatso, what does this look like? And Klatso said, I'm afraid I'm unable to give you any useful leads as far as the etiology, the cause of this disease is concerned. It seems to be definitely a new condition without anything similar described in the literature. The closest condition I can think of is that described by Jakob and Creutzfeldt. And again, nobody listening to this PowerPoint has heard of Jakob Creutzfeldt, or frequently called Creutzfeldt-Jakob disease. But Klatso was a scholar and a pathology scholar. However, in cases reported, the neuronal degeneration was most intense in the cerebral cortex, as in these kuru non-human primates. Also, no cases involving children or adolescents are mentioned. The etiology of Jakob Creutzfeldt is entirely unknown. Only about 20 cases have been reported. But without a moment's hesitation, Gatashek sent out letters to medical centers looking for cases of Creutzfeldt-Jakob and was able to obtain central nervous systems of Creutzfeldt-Jakob individuals. And again, he was able to transmit Creutzfeldt-Jakob to non-human primates. Really a brilliant tour de force. Remember, he was a man who was this incredible explorer in the 1950s, went into this, what we call endo-cannibalistic region. In other words, they eat their own, was able to get central nervous system, was able to take the central nervous system and inoculate it and hold the animals for a while, and then do the same for Creutzfeldt-Jakob disease. So these diseases have a variety of names, but generally now they're called prion diseases. And these prion diseases are diseases with non-inflammatory pathology, no inflammatory cells as you might see in somebody who has acute flu. And they're transmissible. But what's remarkable about these diseases and their transmissibility is that the transmissible agent is unconventional. And why do I say that? So these agents are resistant to physical and chemical reagents that nucleic acid containing agents are usually susceptible. In other words, viruses, bacteria, you can inactivate them. But prion disease agents doesn't seem that way. They seem resistant. And also there's an incredibly long incubation period compared to overwhelmingly other transmissible diseases. And we have a variety of human prion diseases. Probably none of you have heard of these diseases. Kuru, Creutzfeldt-Jakob, Gerstmann-Strauss-Schenker disease, fatal insomnia. Some of these diseases are sporadic. In other words, there's no evidence that they're inherited from anyone, but some of them are inherited, what we would call familial. And in some cases, there are iatrogenic versions. Iatrogenic meaning that the medical profession in some way is actually transmitting these without realizing it. And then there's variant, Creutzfeldt-Jakob. And there's a variety of animal prion diseases. I spoke about scrapie, but chronic wasting disease, CWD, is a disease of deer and elk that's probably started out in the West Coast around Colorado, but has spread east in deer and elk. There's transmissible mink encephalopathy, feline spongiform encephalopathy, a variety of others, and also a disease called bovine spongiform encephalopathy that I'll talk about. So how are these diseases propagated? And so what we think is that there's a normal prion protein, what we call, but that this prion protein is misfolded. In the blue here is PRP scrapie, and we normally call the misfolded prion protein PRP scrapie. And what we believe is the case is that the normally folded PRPC, when it interacts with this PRP scrapie, it gets converted into PRP scrapie, so that there's kind of a systematic conversion here. And this conversion could occur in many ways. You could take Kuru tissue or Creutzfeldt-Jakob tissue, inoculate it into an animal, and you have PRP scrapie that's probably going to convert the normal PRP cellular into PRP scrapie. And this kind of shows it in a way. On the left is the normal structure of PRP cellular. It's resistant to proteases, things that break down proteins. And on the right is PRP scrapie. It's got a different structure. It doesn't have that same nice folding. And it could be infectious, in other words, transmissible, and it's protease resistant. So if one took PRP cellular, there's this little piece of gel there, and added proteinase K, it all gets dissolved. But PRP scrapie doesn't all get dissolved. It's resistant to the protease. And you see here on the right side, when one adds proteinase K to the PRP scrapie, there's a resistant core. And so it's kind of remarkable in a way that that this misfolding causes protease resistance. So as I mentioned, we have inherited prion diseases. And these inherited prion diseases have a mutation in the gene that causes the normal cellular PRP. And that mutation causes the PRP to be misfolded, which induces disease. And this was established with a landmark paper. Senior author here is Stan Prusner, who won a second Nobel for these diseases. And this is GSS, Gerstmann-Strauss-Schenker syndrome, which is an inherited prion disease. And what Prusner did is he took the abnormal mutated prion protein, a mutation that causes GSS, and he put it in a mouse. He makes a transgenic mouse. And this transgenic mouse turns out to develop Gerstmann-Strauss syndrome. Kind of a remarkable experiment. So just to summarize things at this point, PRP scrapie has the same amino acid sequence as PRPC, but a different conformation, a different folding that enables it to be protease resistant. PRP scrapie is present in all prion infections. Sometimes it has the same amino acid sequence, but it's misfolded. It accumulates in prion disease, and I showed you it converts PRPC to PRP scrapie. And that PRP scrapie is a proteinaceous infectious agent, what we call a prion. And PRPC is mutated in familial prion diseases like that Gerstmann-Strauss or familial Creutzfeldt-Jakob. And that mutated PRPC predisposes it to be converted to the misfolded PRP scrapie form. And this shows one other small important feature of these diseases. And the important feature is that you kind of need something with a similar species. In other words, if you have PRP scrapie from a human and inoculate a mouse or a hamster, it may not work. In other words, the PRP cellular of the mouse or the hamster may not get converted to PRP scrapie with the human PRP scrapie. You may need hamster scrapie PRP or mouse scrapie PRP, or a number of inoculations to sensitize that animal that's from a different species. I mentioned iatrogenic Creutzfeldt-Jakob, and this is, of course, one of the tragedies that occurred in which people transplanted tissues of one individual to another individual. In some cases, for example, this Duramata graft, Duramata is a lining of the brain. The Duramata graft was in a mixture of Duramata grafts, a bunch of them. And so if one is infected and you have 100 grafts in there, you're going to have contamination of multiple grafts that are going to be inoculated into the individuals. And so we're very careful now. And I think if this occurs, it must be extremely rare. I alert you to the median average incubation period. And you see here growth hormone. Now we have recombinant growth hormone. In other words, it's not taken from a human. We have some DNA and make this growth hormone. But in the old days, we had a pituitary, and we had a bunch of pituitaries and extracted the growth hormone. There's a 12-year average incubation period. Kind of incredible, the incubation period of transmissions of these iatrogenic Creutzfeldt-Jakobs. One of the important issues here is like many genes, DNA makes RNA makes protein, there's a little variability. And one of the variations is one of the amino acids, amino acid number 129, and whether it's, remember there's two genes, and so you really have two amino acids. So if you have methionine, methionine, or methionine, valine, or valine, valine, makes a big difference with respect to transmissibility, probably having to do with the folding of this PRP. So the other rather frightening event that happened in the case of Prion disease was mad cow disease, or bovine spongiform encephalopathy. And this shows the first case of BSE back in the 1980s. A feed ban was introduced, but really there was an enormous numbers of these are cow cases that occurred, enormous number, back really into 2000. And what we know now is that tissues from sheep probably with scrapie, contaminated cattle feed in the 1980s. And this cattle, maybe the sheep, caused BSE in the cattle, and the prion-infected slaughtered cattle were used as cattle feed. So they were continuously recycled back into the feed, really getting a pretty good level of the infectious agent. And the slaughtered cattle, everything was used in the slaughterhouse. And they have a process called rendering, which was changed during the early years of BSE when nobody really knew there was BSE. And this rendering procedure was changed so it was very efficient, very quick. As a result of that, it allowed maintenance of sufficiently high prion levels for efficient transmission. So watch out for some of these manufacturing changes that make everything move along quickly, because safety may be an issue here. In 1995, the Minister of Health in the UK said, quote, "There is no conceivable risk of BSE being transmitted from cows to people." And here he is eating what they call a wimpy burger with his granddaughter. And this is December 3rd, 1995. And here, March 21st, 1996, Robert Will, who was a leading figure in the UK, and his quote, not so long after the prior one, "In the past few weeks, we believe that we may have identified a new clinical pathological phenotype of Creutzfeldt-Jakob, which may be unique to the UK. This raises the possibility of a causative link between BSE and CJD." And in fact, they were linked, no question. And variant Creutzfeldt, it had a slightly different clinical picture because it affected younger individuals, and the pathology had a few little tweaks, but it was Creutzfeldt-Jakob. But variant emerged at a similar time to BSE, co-localized it geographically with it. In other words, there's very little BSE in the United States, but it was very big in the UK. And transmission of BSE or mad cow or variant Creutzfeldt to experimental animals resulted in similar pathology. And the protease-resistant prion was very similar. So variant Creutzfeldt was probably orally transmitted to humans from BSE-contaminated food. Now, remember I told you that there's this kind of species block. But I also mentioned that cattle feed, probably prion-contaminated, had been continually given back to the cattle. So I think this was very high-titered, very high level of infectious agent. And maybe the species barrier, it isn't always as strong as one might think. And this shows this BSE and variant Creutzfeldt. What does it mean? Well, number one, you could see that probably the incubation period here was a little bit long, but it's scary. Fortunately, We have no variant Creutzfeldt-Jakob now. And we have no mad cow, as far as I know, but maybe there is a little bit. This says something like, is there mad cow in the US? And that's my little joke, except at branding time. And scrapie. Be aware that scrapie in sheep certainly occurs. Now, farmers are aware of it and certainly know that they must be slaughtered or call in the health individuals. This is 1977, but scrapie's still here. And the other is CWD, as I mentioned, which is a prion disease of elk and deer and farmed elk that are used for food. And this is a little bit of an old map, but shows CWD in free-ranging populations. Originally Colorado, but it's spread certainly to the Midwest and also to the East Coast. I'm not sure about the West Coast here. Now, we haven't had CWD transmitted to humans. Maybe that species barrier is here, but maybe it's got a long incubation period, a little bit unclear. So these are non-inflammatory diseases, no inflammation, and looks like an inherited disease, autosomal dominantly inherited, kind of like Huntington's disease or other kinds of autosomal dominant diseases, but non-inflammatory diseases and familial diseases can be transmissible. The other thing is, remember, this is an unconventional agent, prions. Unique properties that can be active in human neurological diseases. These are protease-resistant proteins. As far as we know, they don't have any nucleic acid like viruses or bacteria, but they're transmissible. The third kind of important thing is diseases of animals and diseases found in exotic areas can provide important clues that change our thinking regarding medicine and science in general. So remember that the clue about these diseases came from a veterinarian neuropathologist, Bill Hadlow, who was studying scrapie in sheep. And remember, this first Nobel Prize for prion disease was Carlton Gattishak, who was investigating kuru in the foray population in the highlands of Papua New Guinea. So a lot of obscure kinds of diseases may have very important impacts. So what kinds of questions do we have? So one is, how about sporadic, non-inherited Creutzfeldt-Jakob that occurs worldwide? What causes this? Does it have to do with ingestion of a prion disease animal? Are there other things that we're not paying attention to? That's still a very big question. Another one is, what other non-inflammatory neurological diseases may be caused by misfolded proteins? And the answer is a lot. Alzheimer's disease is caused by beta amyloid. That's a misfolded version of amyloid precursor protein. ALS is associated with misfolded TDP-43, ALS being amyotrophic lateral sclerosis. Spinocerebellar degenerations have misfolded proteins. And a lot of misfolded proteins may be involved in many, many, many diseases. The unfortunate aspect is that we have no treatment for these diseases. We have no treatment at the moment for prion diseases, and we have no treatment for many neurodegenerative diseases that are caused by misfolded proteins. So why do I present this PowerPoint to the group here? Maybe for a variety of reasons. Number one, try to be fortunate and get a mentor who's going to guide you, who's not going to take advantage in any way of you, but is going to help you make career decisions that you're going to be happy with. Second, watch out for misfolded protein diseases. Remember, Gatashek's letter goes back to the 1950s, but in fact, misfolded proteins are still very, very important in science, in medicine. And the third really important issue is treatment of these diseases. How are we going to refold a misfolded protein? Now, we have chaperones in our body that refold, but we haven't been very successful with the prion diseases. Anyhow, I've spoken for a long time. Nina, do you want to take over again? And I'm happy to answer any questions, or maybe you want to, whatever.
Yes, wonderful. Awesome. Well, we do have a few questions in the chat from our students. I'd love to also mention again, if anybody has any questions, please feel free to type them in the chat. If you are interested in communicating directly with our professional today, you may also use the raise hand function under the reaction button on Zoom and raise your hand, and then you'll be able to unmute yourself upon being called and ask your question directly. So the first question that we have here in the chat is that you mentioned that prions are protease resistance, but is the immune system response, like macrophage phagocytosis, to be activated to remove these proteins?
That's a good question. Remember that PRP scrapie is actually the same amino acids as PRP cellular form. And we don't make an immune response against our cellular PRP. Now, it is true that it's misfolded. When I said that there's no immune response, it depends on what you mean by an immune response. We do have these cells called glia, and these prion diseases have a glial cell response, but in no way is there any reaction that mitigates the death and destruction that prion disease has. Is that because the amino acids are the same, or the misfolding isn't enough to trigger a good immune response? I don't know the answer to that. A number of people have spent a long time looking at, is there an immune response? How can we make an immune response? But we're still a little bit in the dark. The other thing I want to make clear is I showed PRP cellular and I showed PRP scrapey. Is there something else in this infectious agent that we haven't yet clarified, identified? And the answer is I don't know. These are difficult diseases to work on. Remember, the incubation period is very long. So, you know, how are you going to get funding if it's going to take a year or 18 months? And you want to have a very safe environment, because you don't want somebody who's taking care of these animals to get bitten and come down with prion disease. So it's an expensive investigative pursuit, for sure.
Thank you so much, Dr. Roos. Our next question is from Sonia. Is there a way to test for prion diseases, for example, by testing for the proteins before they actually manifest? She says she asked this because her grandmother died of CJD 1718 years ago. And is there anything that we can do at all to destroy them, either innate or foreign?
What was the last question? I missed that. What?
The last part of that question was, is there anything that we can do to destroy them?
Oh. So I didn't talk about testing, and we actually have a very sensitive test for prion disease. Basically, it's a little bit like that slide in which I showed normal cellular PRP gets converted to PRP scrapey when you provide a little bit of PRP scrapey. So this test involves adding a minute amount of PRP scrapey to spinal fluid that one wants to test. And one keeps on doing this. And if the spinal fluid has PRP scrapey in it, it becomes amplified. And one could identify that that spinal fluid has prion in it. It's very sensitive. I'm not sure that there's a way to test for prion disease without having central nervous system tissue or spinal fluid. Now, in animals, one has prion disease a little bit more systemically, and one could look at other tissues like tonsil. But I don't think that that's been efficiently done in humans. I might also say that my talk was about prion disease and the prion diseases that I spoke about. But I also mentioned neurodegenerative diseases. And there are a number of investigators who are looking very carefully at these neurodegenerative diseases to see whether there's like a prion state there in which some protein misfolds. Kreuzfeld, there's kind of a stochastic change in the whatever. And maybe those bit more difficult to identify that misfolded protein. Maybe it's a bit more difficult. At any rate, you have a grandmother that died of prion disease. It would be good to look at some tissue. Probably it's a little late now, to see whether there might be a mutation of PRPC. In other words, could this be familial prion disease? Probably not, if there's nothing in the pedigree. There certainly are some very, very, very large pedigrees of prion disease, Creutzfeldt-Jakob, multiple generations going back. Yes, Nina, sorry, I'm dribbling on.
No worries at all. Thank you so much for your response to that question. Our next question is from Connor. Connor asks, do you still see patients in a clinical setting? If so, what are some common or interesting conditions that you typically see in the clinic?
So I may have missed a little bit of that, but I think they were asking about moi, right? Me. Am I right? And I am director of a amyotrophic lateral sclerosis clinic, ALS being motor neuron disease. There are inherited ALSs, maybe 5 to 10%. And I study one of the genes that gets mutated in inherited ALS. Very interesting mutation. We have about over 30 neurological diseases that have expanded nucleotide repeats. So, for example, in Huntington's, there's 3 nucleotides that get repeated. In ALS, the most common cause of inherited ALS is C9orf72, which has a hexanucleotide, GGGGCC, GGGCC. And most people have about 10 repeats. But in C9orf72, there are hundreds or 1,000 repeats that vary within one individual. And we have over 30 expanded repeat diseases in neurology. So I'm interested, especially in inherited ALS. I might mention that there are a number of variants of Creutzfeldt-Jakob, and one of the variants is the amyotrophic variant of Creutzfeldt-Jakob, which looks very much like ALS initially. I wouldn't rule out the possibility that the whole phenomena of misfolded proteins is very important in ALS. I mentioned that virtually all of ALS has TDP43 that's misfolded. At any rate, I have had a very satisfying career, mentored initially by Stanley Aronson, going I've been very fortunate, and I hope the people that are listening Nina?
Yes, I'm here. I
Sorry.
think my connection dropped out for a moment.
Oh, yeah, I got scared.
No worries. Our next question for you is, did you ever face doubts or not have support from your family or friends on your path to becoming a doctor?
Did I ever face doubt from my family and friends that I missed the end?
Yeah, sorry about that. I think I'm having some Wi-Fi connection issues. The question is, did you ever face doubts or not have support from your family or friends while on your path to becoming a doctor?
So my family loved doctors. I had an uncle who was a GP on Long Island, and he was much loved by the community. I had another uncle who went to medical school in Edinburgh, but the war broke out and he became a psychologist rather than a physician. My father was on the board of trustees of Maimonides Hospital in Brooklyn, and physicians were hugely respected. And so I had wonderful support from my family, and friends. Friends are your friends. They're not going to say, "What do you want to do that for?" or something. They seemed to be happy with what I was doing. You have to follow your spirit, what you want to end up doing. I was fortunate in many ways. I'm sure there are some people on the call and the family wants this or that to do. My wife's father always wanted to be a doctor, but there was no money, and he ended up having 3 jobs, and that was the way it is. So sometimes other environmental factors trump what you really want to do. And hopefully you get satisfaction from life, whatever you do.
Thank you so much, Dr. Roos. Our next question for you is, how much of your time is spent with patients versus doing administrative work?
How much you spend in patients versus what?
Administrative work.
Oh, well, I actually was chairman of my department of neurology for almost 10 years, but that's not true anymore. And I don't have a lot of administrative work, but science is important to me. I run a laboratory. The main focus of my laboratory is C9orf72, the gene that causes most cases of inherited ALS. And I have a number of collaborations. One of my collaborators works on C. elegans, which is the scientific name for a small worm. And we have transgenic worms in which we put C9orf72 with the expanded repeat in the worm. And the worm actually comes down with a disease. We also have some collaboration with a scientist who has what we call IPS cells, induced pluripotent stem cells. And these are cells, for example, taken from somebody who had ALS from C9orf72. A skin biopsy is performed, and the skin is treated with a certain formula of reagents that brings them back to stem cells. In other words, cells that could become many different types of cells, like a skin fibroblast. But these IPS cells are then treated with other reagents that makes them central nervous system cells. So IPS cells are very important because they provide the possibility for investigations in vitro. In other words, in a flask, in a petri dish, rather than in an animal. It's less expensive. It's human. These are human IPS cells. And you could always criticize, what a worm or a non-human primate or a mouse, is that really the same as a human? So IPS cells are very valuable. They bring things back to the human.
That's very interesting. Our next question is from a student. what advice would you give to aspiring doctors interested in neurology? And for any aspiring doctors who aren't sure what they want to do yet, what could you say to persuade them into considering neurology?
So neurology is a wonderful field, no question. And it used to be kind of a little bit of a joke in which somebody had a neurological disease, but there was no treatment for this individual. Of course, that's very tragic. But we have huge technology now. And I mentioned spinal muscular atrophy, which was essentially a motor neuron disease like ALS. SMA, most cases are in young people. 95% of kids were dying before age 2 of SMA if they had mutations. Antisense oligonucleotides now save these kids that are making milestones. So here's a disease that was extraordinarily tragic that now is treatable. It's incredible. So neurology is a great field. We have the possibility of treating Alzheimer's, stroke, and many other neurological diseases, epilepsy, and a whole spectrum of abnormalities, both with drugs as well as sophisticated technology. Strokes, when I originally became a neurology resident, there was really nothing to do, almost nothing. And now we could basically stop the stroke from occurring and bring the person back. So it's a great time for neurology. It's a complicated field, but it's a very satisfying one, either as a clinical person or as a scientist or both.
Thank you, Dr. Roos. What would you say is the hardest part about your day-to-day job?
Wow, that's a hard question too. Well, I think, you know, I presented, I'll give you two examples. One was I presented what I think is a fantastic story about the success of science. You know, it's just phenomenal that somebody stumbled into this Okapa in New Guinea, and we ended up having 2 Nobel Prizes and the importance of misfolded proteins. But science sometimes is very frustrating, and many of the experiments don't work. And if that's going to discourage you, that may be an issue. On the other hand, some things work. And when some things work, it's great. So yeah, having lack of success sometimes is discouraging. And the other thing that's discouraging is making diagnoses for patients that you're unable to treat. ALS causes death usually in 3 to 5 years. And at the moment, we really have no effective treatment, although there are many, many clinical trials out there. And so that could be discouraging as well. On the other hand, you're helping people. Even if you're not preventing their death, you're providing a very important person for the patient and the family. And so that's kind of how one has to balance the lack of penicillin for pneumonia for these neurodegenerative diseases.
Thank you very much. Our next question for you is, can you speak to any of the biases that you feel like may be hindering your profession in any way? And how do you go about dismantling these biases in the favor of truth?
That's a really good question. I think what's important here is why Gattashek won the Nobel Prize. And he won the Nobel Prize because people didn't think that inherited prion diseases or non-inherited prion diseases that have no inflammation, that go on for months or incubation periods for long periods, are transmissible because of the bias. So Nobel Prizes are frequently won because the bias is shown to be incorrect. Prusner basically showed that the agent here is a protein, a protease-resistant protein. Again, you know, if somebody had said 50 years ago, oh, this protein causes a transmissible disease, he would have been laughed out the door, okay? Just because of biases. So watch out for that. Some biases are absolutely correct, but it's always important to challenge thinking that's accepted, because maybe there's a bias there.
Wonderful. We have a question from a student in the chat. What advice would you give to those who are actively researching neurodegenerative disorders in terms of publication and lab research?
Wow. So it's a tough question because I'm not exactly sure of the status of the person who's asking this question. Sometimes it's important for somebody to do some research to see whether they're challenged by it, whether they enjoy it, whether they have ideas about it. And publication may not be so important. On the other hand, I have an undergraduate who was in my lab who was very mathematically oriented. And he had all these algorithms to look at neurological repeat diseases and try to figure out some aspects to these diseases. And he's applying to medical school. And in his case, I think it's very important that he get a publication. I think it means a lot, especially because he's all involved in machine learning. So it's a tough question because it depends on who you are, what your rank is, what you want. Sometimes getting a publication isn't nearly as important as being involved in something that gives you satisfaction.
Thank you so much. And kind of following this idea of research, how would you recommend individuals who are interested in getting into research, how do you recommend they go about that?
Another tough question. So if they're an undergraduate, if they have a mentor, the mentor may be able to guide that individual. And maybe in the college, there's somebody doing research that 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 individual might be excited about. If they're a high school student, it gets a little bit more complicated. Again, I think they probably need some mentorship, somebody to guide them. It's interesting, David Baltimore won the Nobel Prize for many things, and he did research in high school, I think at the Rockefeller, maybe I'm wrong. So there are opportunities, you know, it depends on where you live, what institution is available. One of the problems, of course, is that if you're in high school, you don't have a lot of time, you don't want to work all night, you don't want to work on the weekends and so forth. So there's some conflicts here that have to get resolved. So it's not easy for me to answer that question, not knowing who the questioner is, but it's an important question with very important answers, no question.
Wonderful. So I'd like to open this opportunity. Are there any students that are joining us for the live virtual shadowing session today who are interested in raising their hand and asking their question directly to our professional today? Now is your chance if you are interested in making that connection. If not, no worries. Feel free to continue typing your questions in the chat. We do have another question. Dr. Roos, do you ever experience burnout?
That's a tough question. One of the challenges with respect to lab work is money. These lab experiments are expensive. Biotech companies have a lot of money, but if one's working in a medical center doing investigations, one needs money. Sometimes a lot of money. Prion disease, you know, I mean, in order to house non-human primates in a primate, you know, a lot of money. But even worms or mice or whatever you do. And so one ends up writing grants. The big funder for grants is the NIH. But you have to get accepted. And it's not easy to get accepted. And acceptance depends on a committee that reviews you. And they may not be on the same wavelength as you, whatever. And so burnout is a problem if you don't have enough money. I have been fortunate of late that I've had some donors that have helped me hugely. But it's hard to get donors in the beginning. It's a challenge. And so then one needs grants. Now, what's nice is the NIH likes to support early investigators. That's good. But nevertheless, there's a lot of competition out there. I also have to say that some medical schools are very interested in research. They're the great medical schools in the United States. But the overwhelming majority of medical schools don't want you to do research. If you're a medical student or go into medicine, they want you to see patients, make money, blah, 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 blah. So there's a little bit of a conflict here that one has to resolve.
Thank you so much, Dr. Roos, for your time today and for sharing this insight with us. I found your presentation to be very interesting. I learned a lot about prions personally, and I'm sure that a lot of our students did here today.
Okay. Are you going to have venison tonight?
I will have to see, potentially.
All right. Take care. Thanks,
Thank you.
everyone.
Thank you
Bye-bye.
so much. Students, do stick around if you're interested in figuring out how to earn your certificate with pre-health shadowing. So before we hop into that, I always encourage a nice reflection. Just because you may be using virtual shadowing sessions with pre-health shadowing for your application when you are applying to your professional programs. So it's always great when gearing up to submit your essays and personal statements, just a little bit about what you learned and what you took away from these sessions. So here are just some 3 questions to get your brain going. That being said, if you want to utilize this reflection for more than just your essays, we also do have opportunities to get published through pre-health shadowing. So if you're interested in having a featured publication, which you can include on your resume, CV, your LinkedIn profile, and more, I definitely encourage you to check out our website, prehealthshadowing.com/blog submissions. We take a bunch of different pieces of work from the pre-health shadowing community and spotlight and highlight our students that are doing amazing things. Earlier I mentioned being a part of the pre-health shadowing program. If this is something that interests you, we are taking applications now for the pre-health shadowing leadership team. You can work with various pre-health students around the world in a bunch of different fields. If you're interested in project management, we have opportunities there, grant writing, fundraising, as well as media creations. You know, maybe you have a passion for creating videos, even TikToks. And so if you are interested in any of these, we have a bunch of different opportunities. Check out our website and view the application form and have an opportunity to interview with a member of our student team. If you don't have that much time, that is totally okay, but you still want to get involved in pre-health shadowing, you can sign up to be a volunteer. Volunteers are able to participate in a lot of different tasks to help keep the organization moving. So definitely check out these links if you're interested. Again, please scan this QR code to donate. If everybody here donates $1 to $5, we'll have enough money to keep our website up for the next month, and that would be extremely helpful for those that are working to get virtual shadowing hours through the ongoing COVID-19 pandemic. As I mentioned, if you're unable to donate, please share this link. You can post it on social media. You can send it to your friends, family, and community. We really do appreciate your support in being able to promote our mission of fighting inequity in health education and promoting diversity in the various healthcare fields. So thank you. Now the time we've all been waiting for. If you're looking to earn your digital certificate, this is how you do so. You can go to the website, www.prehealthshadowing.com, and find Dr. Roos's page on our website. His profile page will open up. You can get past the presentation portion, which you are currently on, and it'll open up the quiz. For the quiz, you will have 10 questions, and you'll have 30 minutes and 2 tries to get a 70% or higher. Upon achieving a 70% or higher, you will be granted a downloadable certificate with your name, the date, and the name of our professional. And you can utilize this for your future applications. If you missed any part of the session today, no worries at all. We do post all of our virtual shadowing sessions online on YouTube and our website completely free of cost for your review so you can gain insight. Be sure to catch our sessions every week. We do have an amazing lineup of professionals. Be sure to check out the pre-health shadowing website, sign up for our email list, follow us on social media, never miss a session, get your hours, and participate in the org. Thank you everybody for joining. That marks the end of our virtual shadowing session today. Feel free to disconnect if you are all done. If you have any questions about joining the pre-health shadowing team, I'll stick around for a few minutes to answer those questions. Thanks everyone.